Hi guys, welcome back to our ongoing discussion of biochemistry and biophysics. Now, over the last several lectures, we've been spending a lot of time talking about protein structure and protein stability. Took a segue into protein folding and some of the thermodynamics and some of the dynamic aspects of protein folding. Took another sidebar into thinking about the interaction of light with matter, and now we're going to start putting some of these ideas back together as we turn our focus now to trying to understand how these beautiful and intricate three-dimensional structures of proteins specify their function. And remember, that's one of our top line goals, right, is understanding how structure and function are related to each other. Now, if you'll remember, one of the things I started out pointing out to you guys is that when you think about specifically protein function, right? I kind of like to categorize this as this sort of three-layered, can go more than this, but sort of three layers we're going to get to building connections of function, right? Where we started at the lowest level, other than being structural, the simplest thing that a protein can do, I pointed out, right, was bind stuff, right? And that's where we're going to start, what we're going to start thinking about today. But then I also pointed out that binding, once we understand how proteins bind things, that leads us then into an ability to think about catalysis, how proteins carry out the acceleration of chemical reactions, right? And what we'll see when we get to it is that catalysis in many ways is a really elaborated and very specific type of binding that proteins carry out on specific molecules, right? And then finally, there's this sort of next layer up, we have this idea of energy transduction, right? Where proteins use their ability to bind other things and interact with small molecules that are serve as sort of energetic power sources to then do useful work, right? They essentially take binding and chemical catalysis, put them together to make things happen, like that listeria motoring around in that PTK2 cell we started out with, right? Okay, so we're going to start out, as I said today, walking down this cascade of protein function by beginning to think about binding and binding phenomenon. And specifically, or maybe another way to put this, what we're going to be talking about, something known as protein ligand interactions, right? Okay. So the as I mentioned, once again, binding stuff is one of the simplest things that a protein can do, right? From my point of view, of the way we're going to be thinking about this, I like to break this down into sort of two general areas of thinking about binding. And the first are sort of characteristics of the kind of binding we're going to be thinking about. The first is this idea of specificity, or put it another way, discrimination. Right? And if you want to know what I mean, if you think back to that example I gave you guys with that protein that runs that glucose 6-phosphate reaction we haven't talked about yet, and I pointed out that that protein is really good at discriminating between ATP versus GTP, right? And it essentially won't use GTP, right? But it'll use ATP, right? Depending on the isoform of the protein, right? So that ability to pick out an incredibly fine level, you know, just a one or two or a few atoms different in a molecule that's otherwise the same, and then recognize it with very high specificity is a key characteristic we want to think about and try to understand. One of the other things that's really important to the protein ligand interactions that are relevant across so much of of biology, and is the idea of reversibility. All right, so the kind of binding we're going to be thinking about is fundamentally reversible binding. We're not talking about sort of one-way covalent bond formation. We're talking about binding that is fundamentally reversible, which means we have to think about binding as an equilibrium phenomenon. All right. And this reversibility is also key to understanding how binding phenomena are linked to control, how these ideas of reversibility and the equilibrium associated with that allows different protein ligand systems to impose control on the assembly and disassembly states of biomolecules, on catalysis, on energy transduction, all kinds of stuff like that. All right. So the ideas here that we're going to develop undergird at a fundamental level all the basic sort of protein ligand interactions that happen, and in a more general sense, other types of biomolecule ligand interactions. But we're going to specifically restrict ourselves to protein ligand interactions. And the example we're going to use to build up this set of ideas specifically is the example of oxygen binding by proteins known as the globins, which is a large family of proteins, right? Which I know you guys have heard of. I have two really important members, so-called myoglobins, which in shorthand are usually written capital MB, all right? And the hemoglobins, which in shorthand are usually written capital H, lowercase b, right? So what we're going to do over the course of the next few lectures is build up a formalism that can quantitatively describe oxygen binding to the globins, right? Both myoglobin and hemoglobin. Things are relatively simple when we start thinking about myoglobin, and they get quite a bit more complex when we start talking about hemoglobin, but in a really fascinating way, right? And again, like I said, these principles are going to apply to at a base level to just about any protein ligand interaction you would come across, right? Things can get more complex, and we're gonna touch on some of that layers in which that complexity can develop. But the basic ideas hold across a huge space of protein ligand interactions, right? Okay, so a reasonable place to start then is, well, why, right? I mean, why this problem? Why use this one? Why oxygen binding to the globins, right? Well, for one reason, it's essential, right? Without oxygen and its a role that it plays in our metabolism and the metabolism of every aerobic organism in the biosphere of this planet, there'd be no life as we know it, right? There'd be life, but not like we know it, right? So, and oxygen is just essential for all kinds of basic stuff. Like I said, basic metabolism, which you guys may or may not remember, basic aerobic metabolism depends on it. There's another recent example. Remember the idea we've talked about of disulfide bonds formed between two cysteines in a protein structure? Well, think about it. You synthesize a protein, it's got a couple of cysteines in it. They're reduced when it comes off the ribosome. You want to stick them together, you got to get rid of a couple of hydrogens, you got to get rid of a couple of electrons. Where do those electrons go? Basically, you need to oxidize those two cysteines to form that disulfide bond. Where do the electrons, where do the spare electrons go? Turns out the ultimate acceptor in the large, probably not all, but the majority of organisms, the acceptor for those oxygens, where they get dumped, those electrons, they get dumped in oxygen, right? And there's a huge number of, you know, the list goes on and on and on of other examples, all right? All kinds of molecules you can't make without oxygen, right? Okay. There's another reason we're going to start and we're going to use this as an example, is because the globin oxygen binding system is so well behaved, right? And so it's been so well characterized because it's so well behaved. It may be one of, or maybe the best understood example of a protein ligand interaction across a huge range of domains: structural, biophysical, biochemical. It's just, it's an incredibly well understood system. So it's a good place to start for trying to understand how this works. Okay, a little more context setting. This system, this binding of oxygen by the myoglobins and the hemoglobins, is designed to solve a particular problem in basic biology of organisms like ours. And so the question is, you guys know what that is? You familiar with the problem? There's a fundamental problem in an organism built like we are, that depends upon an oxygen-based metabolism to do all this cool energy production and all the rest of this stuff. Solubility, right? You got a big problem. Oxygen is just not very soluble. In water, like at all, right? And you got to get oxygen to these peripheral tissues deep inside an organism like us, not going to diffuse across our skin. And because it's not very soluble, we need roughly to exist at our size, and with the metabolisms we've got, we need approximately 100-fold concentration of oxygen in our bodies compared to the partial pressure of oxygen at 1 atmosphere of pressure, right? That is a lot, right? So oxygen really just isn't that soluble to begin with. We need 100 times more than would normally be there at sort of atmospheric pressure. How do you solve that? Well, that's one problem. It gets worse. There's an even worse problem, right? What do you know about molecular oxygen in terms of, things it can do? It's really reactive. You've heard of things getting oxidized, right? Well, that's because molecular oxygen can be extremely chemically reactive. Having molecular oxygen at 100 times the concentration freely roaming about in an organism like us would be just death, right? Right? You'd be completely, there's none of the chemistry you'd want to run or proteins you'd want to have would be able to exist with free oxygen running around at 100 times atmospheric pressure, right? You'd just be impossible, right? So we got to come up with a way to solve that problem. All right, so we got a solubility problem. We got to keep the stuff under control. How do you do that? Well, it turns out part of the answer to this problem is that low oxidation state metals for example, iron, copper, a few others, are really good at binding oxygen, right? These guys will bind oxygen really well and interact with it, right? But they're not very soluble either, right? Somewhat, maybe a little bit better than oxygen. So you just shifted the problem, right? You haven't really solved it by just like having a metal oxygen complex, and there are other problems because the oxygen will oxidize the metal and then it won't bind anymore. So that's not going to work. Okay, so here's a potential solution. This is one evolution came up with, pretty clever. Turns out there's a relatively small organic molecule that you can use to hold these low oxidation state metals like iron and copper and manganese and other things, right? And it essentially involves a structure that looks like this, where you basically take a parole ring. If you remember, a parole is this 5-membered ring with a nitrogen at one vertex, right? It's gonna have a carbon double bond in it. It's in conjugation around this ring of 4 of these guys to create a so-called tetraparole that are linked up with these vinyl groups, right? And they can have different substituents around the ring. This tetraparole structure positions these nitrogen groups at the perfect distance and orientation in what's essentially a planar configuration of those nitrogens and their pair, non-bonded pair of electrons for a perfect coordination shell for metal atoms like iron and copper and manganese, a few others, right? So this is a great way to hold on to the metal. The other thing that's really cool about it is, for example, iron doesn't want to just be tetrahedrally coordinate. It wants to be octahedrally coordinate, which means that iron bound inside the center of this tetraparole has 2 additional ligand positions or binding positions for something else that can interact with available electrons in that complex electron configuration of a metal like iron. And there's one above and one below the iron if these 4 sort of lateral positions are occupied, to generate a so-called octahedrally coordinate iron. And these are both available and both want to be bound. So that's cool, right? Because now, oh, cool. We could get X, we could bind an oxygen there, right? And we could bind an oxygen there, and we can transport that around, right? And there are 2 problems. One, take a look at that molecule. How soluble do you think that is? Well, this particular one, this happens to be known, or these iron-bound tetraparoles are known as porphyrins, right? Yeah, you got a carboxylate, here it comes, but if you look at all of this, man, look at all, that's pretty daggum hydrophobic. That's not very soluble. So even binding oxygen to this by itself, that doesn't solve the solubility problem. You can't get this nearly concentrated enough to get that 100-fold enhancement. And if we just let these oxygens sort of bind here by themselves, you know what they're going to do? They're going to rip away an electron. That's going to turn into iron 3, which can't bind oxygen anymore. So if you do that, you now lose the ability of that porphyrin. It'll still bind the iron in the tetrahedrally coordinate positions, but it doesn't want to bind things in the octahedral positions anymore. All right, so that doesn't work. So we haven't solved the problem. We sort of took a step, maybe, have to see it, but we got to do something better. And of course, evolution's answer to this problem was to take this partial solution, kind of hydrophobic, not very soluble, and we're starting to build something we can use to hold on to an oxygen pretty well and encase it in a protein, right? And that protein is, of course, for our purposes, as the globins. This is the monomeric version of the globin called myoglobin. You can see it's an almost completely alpha helical protein, right? And it happens to have over here on one side a nice little hydrophobic pocket. It's not totally down in the core of the protein, sort of part way with an edge sort of exposed near the surface, but snuggled down in a nice hydrophobic pocket. And you can see that a little bit, if we put this thing into rotation, how it's tucked down to this nice little hydrophobic pocket, right? And it turns out that's a really awesome way to bind to an oxygen molecule. Okay, well, that's cool. That's really kind of cool. But before we get to the details of how that works, right? Let's think about the biology of this just a little bit more, right? This solves one fundamental problem. By encasing this guy, we can get the solubility up, right? That guy's hydrophobic, it's in the hydrophobic pocket. We surround that, as we know, with a globular protein by putting nice polar and charged stuff on the outside of the globular protein. man, we can probably crank that concentration up quite a bit. We absolutely can. Easily solve the concentration problem. How do you bind the oxygen? Well, we're going to come back to this in some detail, but turns out in that little pocket on the myoglobin, one side of that octahedral position is occupied by histidine, which it now takes care of one of those octahedral positions, right? The other is now open to bind an oxygen molecule, right? And this happens between a couple of positions on a pair of the helices. There's that one heme on the so-called proximal side, and then the opposite, the distal heme, and there's the little pocket between the distal heme, or the distal histidine, I'm sorry, it's another histidine on the other side, and there's a little pocket that's just perfectly oriented for the oxygen to interact with that heme iron atom, right? Now, how this works in some detail, we're gonna look at, but in sort of just broad strokes, that's kind of what's going on with oxygen binding to the heme. Okay, myoglobin does this kind of simply. Hemoglobin, it gets a little more complex. But to just sort of, again, set a little more context here, let's think a little bit more about the biology of what's going on, all right? Because there's some really interesting wrinkles to this. If you think about oxygen transport in an organism like us, and you write down the simple scheme for what's happening, we know we've got oxygen that's going to bind to hemoglobin to form an oxygen hemoglobin complex, right? Probably remember that, at least from some of your biology, right? This then is going to get transported by the blood from the lungs. So this is all happening, let's say, in the lungs, right? This is going to get transported out to the peripheral tissues, right? So the microvasculature, right, in the peripheral tissues, where we're going to have then this hemoglobin oxygen complex that will then dissociate into oxygen and the hemoglobin, right? And now we've delivered oxygen out to those peripheral tissues where we're doing all this cool metabolism that we need to keep ourselves alive, right? Okay, right. So I'll just make this a little more specific, like tissues. Okay. But is that the only thing that's happening? Seems like memory serves, there's something going the other way, right? So oxygen's going out from the lungs, out to the tissues. Something's coming back the other way, right? It's that major league waste product of all this awesome metabolism that keeps us alive. As we burn these hydrocarbons that we talked about in the first lecture to extract these high potential electrons to do the stuff we want to do, we generate a waste product, right? And that waste product, of course, is carbon dioxide, turns out, and water, right? So that CO2 we need to get rid of. That's happening out here in the microvasculature, and it's not transported as gaseous CO2. Some of you guys may remember that CO2 in water will turn into carbonic acid, right? And this normally is kind of slow. Turns out erythrocytes, your red blood cells, are packed full of a certain enzyme called carbonic anhydrase. That dramatically speeds this up. But the whole point is that the CO2 that's being produced as a consequence of peripheral metabolism is turned into bicarbonate and a proton, right? Okay. This then has to get transported back in the opposite direction from the microvasculature and the tissues back to the lungs, where we have now, right, and a proton. And somehow what we need to do is we need to recombine that proton with that bicarbonate, drive off water, to then release gaseous CO2, which we turn around and exhale, right? Okay. But here's the really cool thing. These two events are not independent of each other. In particular, the two things that are really very intimately connected, the binding of oxygen, right, and protons, specifically to hemoglobin, right, is a super important aspect of this process, right? And the first person to recognize this, that something unusual is going on here, right, with hemoglobin, was well over 100 years ago, approaching somewhere in the range of 140 years ago, something like that, by someone known, a person known as Christian Bohr. If you remember a bit of the history of the development of quantum mechanics that we just talked about, you'll recognize that name, Bohr, as in Niels Bohr. Christian was his dad. Christian was a chemist, right? And the father of Niels. And Christian was interested in hemoglobin, right? And understanding it, could tell that it bound oxygen, and we knew this was important for life. People were starting to work this kind of stuff out. And what Christian Bohr figured out was that the binding of oxygen to hemoglobin made it a better acid. So when he looked at sort of the protons released per oxygen bound to hemoglobin, what he found was that at around neutral pH, for every oxygen bound, hemoglobin molecule would almost give up a proton, a little less than one proton, right, for each oxygen bound. So binding of oxygen made hemoglobin a better acid. Another way to think about this, right, is that the binding of oxygen and protons to hemoglobin are antagonizing each other. They both kind of want to bind to hemoglobin, but they can't both be there to the same extent if the other's bound. Another way to describe this is using terminology we already talked about when we thought about protein folding, which is that the binding functions, right, of oxygen and protons to hemoglobin are thermodynamically linked. All right. They energetically influence each other. They're not independent of each other, right? And before we get into sort of an explanation as to why, I'm going to think a little bit about that biologically, because this turns out not to just be some oddball anomaly of the structure of hemoglobin. This turns out to be a functionally, most likely selected property of this system that was encoded over evolutionary time into the behavior of the hemoglobin tetramer. And to understand why, all you've got to do is look back at this scheme, right? Because imagine you're out here in the microvasculature, right? Making lots of CO2, doing lots of aerobic metabolism. And you're making CO2 like crazy, right? You're just pumping this stuff out, right? Which means you're also, at the same time, pumping out, because of carbonic anhydrase, you're pumping out the protons. Okay. But you need oxygen out there because you need to keep this system running. Well, you make more protons as a result of making CO2, the metabolism's doing. Carbonic anhydrase drives that into carbonic acid, releasing protons. The protons can bind hemoglobin And the hemoglobin that's out there has already passed through the lungs and it's oxygen saturated. And as those protons bind, they and the oxygen that's on there can't be on there to the same extent at the same time. So the binding of the protons, out where there are lots of protons out here, pushes the oxygen off the hemoglobin right where you need it. At the same time, this is taking up protons, which helps pull the carbonic anhydrase direction in that direction. So there's a nice little cycle of coupled antagonistic delivery. You take up the protons, give off the oxygen, right? Increase the ability to remove CO2, right? And deposit the oxygen sort of optimally where you need it. And the same thing is happening also in the lungs. If you think about that, you come over here and you think about what's happening over here in the lungs. You transport it back into the lungs, and now you are in a situation where you've got high oxygen content, right? Binding to hemoglobin that's been transported over here and that has a proton bound to it, say. The oxygen is going to bind to that hemoglobin because partial pressure is much higher in the lungs and in peripheral tissues, and it's going to ram itself on there, which is going to drive these protons off, right? What's that going to do to that equilibrium? As the oxygen drives the protons off in the blood in the lung, that's going to drive the recombination reaction of the proton carbonic acid this direction, generating CO2, which can then be expelled right where it needs to be in the lungs, right? Okay, now that coupling, I'm trying to set the context for the biological significance of the coupling we're going to start digging into in a little more detail here, is absolutely critical for the respiratory cycle that our bodies need to run to live, right? And it is significantly based in this thermodynamic linkage between these two ligands, protons and oxygen. There are others, but these are two biggies that make this whole thing work correctly. And the next time we're going to start digging into a little more of the formal detail of how you describe a process like this.